Welcome back to the Basic Bioinformatics Web Workshop. In this lecture, I will guide you step by step through different web and software tools, so you know how to use them for your own research. Before we begin, let us remember what we should have learned from the previous lectures. To perform a sequence alignment, a minimum of 2 sequences are required. Amino acid sequences can be retrieved from UniProt, while nucleic acid sequences are recommended to be found at NCBI. However, both types of sequences can also be found in other databases, like EMBL for example. In addition to your sequence selection, you must choose an algorithm that is appropriate for your intended application. In general, different algorithms must be used for pairwise and multiple sequence alignments, as well as nucleotide or protein alignments. In terms of your research hypothesis, it might be beneficial to determine whether you want to use a local or global algorithm. If you're familiar with these concepts from previous lectures, you should have no problem following the hands-on approach that I'm about to present in this lecture. First, I will show you how to align your sequences in a matter of seconds. After that, we will conduct a database search. And finally, I would like to introduce you to a free software package that you can use for your own studies. As you can see, we will start in our web browser and we start Googling EMBL EBI. And then we press Data Resources. Then we scroll down until we reach Sequence Analysis. Now we should be at EMBL EBI's job dispatcher. It provides integrated access to several bioinformatics applications. If it is your first time around here, I would recommend to read the introduction text. If you have additional questions, make sure to check out the recommended training material. As soon as you're ready, we scroll up again and click on Job Dispatcher. We can take a look at previous jobs. As you can see, there's nothing here at the moment. And as we scroll down one more time, we finally reach the tool categories. Since we want to perform an alignment, we have to select between pairwise and multiple sequence alignment. For now, I would like to start with a pairwise alignment. Here we can select between 3 subcategories: global, local, and genome alignments. As I demonstrated in our last lecture, it is important to know the algorithmic characteristics of your algorithm. So if you're not entirely sure which algorithm suits your individual task, it might be helpful to at least read the caption or conduct further investigation. We will choose the Amboss needle algorithm. It's based on the classic Needleman-Wunsch algorithm that I've introduced in the second lecture. So our choice is quite basic, a global pairwise sequence alignment. First, we select whether we want to align DNA or protein sequences. Beside that, we can choose the output format. You can go through different styles if you would like. Usually, I select the default options. If you click on More Options, you can change the substitution matrix or the gap penalty parameters. Finally, you can name your alignment if you like. Let's name it Test GP1. In preparation of this lecture, I selected 2 sequences which can now be aligned. After getting the results, we can review our settings once again. Then we get a summary on our results, so apparently there should be 2 mismatches and no gaps. If we go further, we can take a look at our alignment and we see that the divergence is displayed within the first few residues. There is a mismatch due to a valine in sequence A and glutamic acid in sequence B. I think the second mismatch might be no mismatch at all, since X in sequence A represents a variable for an unknown amino acid. If you would like to download the result, you can scroll up to result files and download the alignment. That's it for our first example. Since our first alignment was pairwise, I would like to show you how to align a multiple sequence alignment. Therefore, we have to return to Job Dispatcher, click Multiple Sequence Alignment, and select between various algorithms. Once again, and especially with multiple sequence alignments, I recommend to read the description to find the most suitable one for your individual task. Let's just make a muscle alignment for a quick demonstration. Since I haven't prepared any sequences, we will use the example sequences. As you can see, there are 3 different sequences within the box. We name it example MSA and we keep the default settings. After the computation, we can still change the color scheme and highlight certain regions or amino acids. For an overview, I would keep the default cluster 2. With positive and negative, you can zoom in on the sequence, and with the bar, you can navigate through the alignment. However, the alignment can also be viewed in plain text by accessing the tool output section. This can be particularly useful when dealing with fewer sequences. If you like, you can also try out the other algorithms and compare the results for yourself. After conducting alignments at EMBL-EBI, I would like to demonstrate how to perform a database research using NCBI BLAST. Therefore, we use the unknown sequences from the pairwise alignment once more. I've introduced NCBI in my first lecture, so if you haven't seen it, I recommend to check it out. BLAST is a great tool that allows us to determine where an unknown DNA or protein sequence could come from. So you can more or less compare it to Google for genes and protein sequences. There are 4 different types of BLAST, besides nucleotide and protein BLAST, where your query sequence is in the same format as your expected result. BLASTX and tBLASTN gives you the complementary results. So if your query is a nucleotide sequence, you can get a protein sequence or vice versa. In addition to these 4 algorithms, there are some subordinate BLAST tools for very specific applications. If you're interested, you can pause right now and read the captions. The BLAST algorithm can access several libraries, so millions of sequences are considered in the search. If you're interested in the algorithmic behind BLAST, I recommend checking out the supplement of Lecture 2, where multiple sequences are presented that explain the algorithmic details behind it. Regarding our example, we want to use the unknown sequence A from the previous pairwise alignment that we used on EMBL. Since the unknown sequence A was a protein sequence, we click on Protein BLAST. Here we paste our query sequence, sequence A, from the previous pairwise alignment. For this task, we ignore the fact that we could reduce the range of the query sequence. We do not name it or change any of the standard settings. However, we could, for example, choose a specific library, specific target taxonomy, or adjust the algorithm parameters. The majority of searches are more general, therefore the standard settings are just enough to generate a meaningful result. So we have added our query sequence, the unknown sequence A. Now we click on BLAST and wait for our result. Usually it takes 1 minute, but it may take longer depending on the length of the sequence, your internet connection, and the overall traffic on the website. This is what your result should look like. In the top right, you can once again check your parameters. On the left, you can filter your results. I could, for example, type into the filter HOMO, and our results are reduced to 65 sequences. The matches are sorted by eValue, which is a statistical measurement that calculates the likelihood of match, whether it is occurring by chance or actually having a related or inherent connection to the query sequence. The lower the eValue, the lower is the probability that the match is a random match. In addition to the eValue, the identity can be used to select matching sequences. A value of 100% means that the sequence you have entered is a perfect match for something in the database. In summary, the key to successful identification lies in ensuring that simultaneously maximizing identities, minimizing gaps, and keeping the expectation value as low as possible. Beside the table view, you can inspect the results graphically, as an alignment, or in taxonomic groups. Least would not make sense for us since we selected the taxa HOMO beforehand. Usually, selected all is checked. If you only want to compare selected sequences, we click here and select, for example, the sickle cell HBB and go to alignment view. As you can see, both sequences share a mismatch on the 7th residue. If you remember, the same was the case in our AMBL alignment. So both unknown sequences were actually a HBB sequence against its sickle cell disease variant. I hope you now have an idea how to use BLAST for your own research. If you need an alternative to BLAST or want to check your results with another algorithm, I can recommend the Hammer algorithm from AMBL EBI. The handling is just as easy as for BLAST. If you're working with the same set of sequences over a longer period of time, I would recommend using a software tool instead of the online resources shown. This implies that you have saved your sequences on your device in order to save time and effort. In lecture 1, I've shown how you can download sequences. In case you haven't seen it, here is a quick recap. You go on NCBI, search for the protein or gene name. For example, we use HBB and search. Whether you want the protein or the nucleotide, it is up to you. Since HBB is quite a prominent example, we can click here. Now we can either download the FASTA sequence on the top right, or we scroll down to the section Genomic Region Transcripts and Products, click on FASTA, and now we can copy the sequence and paste it to a new text file, for example. It is important to copy the whole text, beginning from the greater than sign, to ensure the FASTA format. Okay, this is how you do it for NCBI. Especially when you're working with protein sequences, I can also recommend UniProt since the website is more clearly structured. So it's quicker to retrieve the sequences sometimes. Okay, since we now know how we can collect sequences, and construct a data set out of these sequences, we can now work with software tools. I would like to introduce you to the software tools Mega 11 and Eugene. Both programs are free, which makes it great for students. First, I want to show you how to make an alignment with Eugene. Therefore, we need to import our sequences first. So you open Eugene, click on Open File, and browse for the sequence files you downloaded. Select the files you want to look at and open them. Now you can decide if you want to open them separately or join them into an alignment. For demonstration purposes, we will choose the latter, so join sequences into alignment. And we click OK. Now a separate window has opened where we can see the sequences. Let's use the slider so we see what kind of sequences we have in our first dataset. This is supposedly a dataset that consists of HBB sequences derived from various taxa. If we would want to align these sequences, we click on Align in the top bar and select our algorithm. All the algorithms are based on HMM profiles, so it is important to research the differences before using them. That's basically the main message from the second lecture. As a quick summary, Clustal Omega is a tool that uses seeded guide trees and HMM profile techniques to generate alignments suitable for medium and large alignments. Klein is more focused on local regions and suitable for large alignments. MAFT uses fast Fourier transformations and is suitable for medium-large alignments. tCoffee is a consistency-based multiple sequence alignment tool that attempts to mitigate the pitfalls of progressive alignment methods, suitable for even small alignments. For our purpose, we are using only 9 relatively short sequences, so tCoffee should be our choice. So let's align. For each alignment, you can change the settings for gaps and mismatches. However, I do not necessarily recommend it for more general tasks. As shown, there are multiple conserved regions within the alignment. The consensus sequence can be seen within the top bar. The darker shade of blue indicates higher similarity. If you do not like the color code, you can change it with right-click, colors, and then there are several other color codes, some of which do not show a degree of match, but an individual color for each nucleotide or amino acid in protein alignments. If a sequence would not fit your dataset, or if you have a duplicate, you can click on the individual sequence and press delete. If you want to add another sequence to the alignment, you can go on Actions, Add, and select the sequence from the files. When you're satisfied with your results, you can click on the 6th icon on the top bar to save the alignment. As you can see, you have several options for file formats. Usually I would just use the FASTA format. We name it test1 and save it in our Eugene folder. If we now close our window and open the file test1, we are back in our alignment. Beside this, Eugene has many more tools to offer. Personally, I use Eugene only for this task because for alignments it is straightforward with its functions and has a clear laid out design. That's what I like about it. But for other cases, I would like to showcase another software. I'm talking about Mega 11. There are several functions that can be used in Mega 11. Many of them are also available in Eugene. The reason why I present Mega beside Eugene is Mega's focus on phylogeny and to present an alternative to Eugene. So if you're working on research that infers evolutionary events, Mega may be the right choice. The aim for such studies is usually to create a phylogenetic tree which shows the evolutionary history between a set of species or taxa during a specific time, commonly displayed in a branching diagram. To construct such a diagram, we have to construct a multiple sequence alignment once again. Therefore, we click on Align and we select Edit Build Alignment. Within the window that opens, we select the first option, Create New Alignment. Another window opens where we have to select whether we are using DNA or protein sequences for our alignment. The training set that I've prepared consists of nucleotide sequences, so I choose DNA. As you can see, the alignment viewer opens. Now we have to add our sequences. Inside the top bar, we click on the icon that displays the white sheet and the red arrow. This allows us to import files from our desktop into the alignment viewer. Usually, you should be able to import Mega, text, or FASTA files. If you should have any trouble importing files, you could also just copy your sequences and paste them into the viewer field. I select my text file, which contains all my sequences, and as you can see, the file transfer is complete and 16 srRNA sequences from 7 different organisms are displayed. The default sequence 1 can be deleted and we are ready to align. With Mega 11, we can choose between two algorithms, Cluster W and Muscle. As already mentioned, Muscle is especially good for medium-size alignments. In our case, it does not really matter what algorithm we choose since our dataset is not too complex, so we just choose Muscle. Now we can review the default settings. I will keep them as they are, so I click OK. After our alignment is calculated, we can check it. If we are sure that this is indeed the alignment we want to work with, we save the alignment. Therefore, we go to Data, click Save Session. Finally, we name the file and save it. Then we close the alignment viewer. Within Mega 11, we now go to Phylogeny and choose the type of tree that we would like to construct. As a quick introduction, maximum parsimony and minimum evolution are methods that try to minimize branch lengths by either minimizing distance or minimizing the number of mutations. The major problem with these methods is that they do not take into account many factors of sequence evolution. Thus, the deeper the divergent times, the more likely they will lead to incorrect or poorly supported groupings. Neighborhood joining or UPGMA are clustering algorithms that make quick trees but are not the most reliable either, especially when dealing with deeper divergence. These algorithms use computational shortcuts that, depending on the settings, fulfill the minimum evolution criterion. Maximum likelihood applies a model of sequence evolution and is ideal for building phylogeny. This is the most commonly used method in publications and is preferred by many reviewers. The main disadvantage of this method was its computational cost, but with today's computers this is not as much of an issue as it used to be. This will become more of a problem with larger datasets. To be honest, the simplicity of our dataset would probably lead to the same result using all different methods. We select maximum likelihood and proceed with the default settings. The results show a phylogenetic tree that groups Escherichia with Salmonella and Pseudomonas with Azotobacter and Azomonas, whereas Thermococcus stands alone. This makes sense because Pseudomonas, Azotobacter and Azomonas belong to the Pseudomonadaceae family, and Escherichia and Salmonella belong to the Enterobacteriaceae family. Thermococcus stands out because it is a species from the superkingdom of Archaea, so it is not even a species of bacteria. It is important to check your results for plausibility after you have computed them through algorithmic methods. Now you could use the menus on the left side to adjust the graphical appearance. I will leave this to your personal preference. Before I end this lecture, there are two things that I want to make clear. First, once again, the reminder that what I've shown you in this lecture should be considered as a first guide or inspiration to use such techniques for your projects or personal studies. However, before applying these methods and presenting the results, it is important to understand how each algorithm works and to be able to justify why you chose the method you did. Second, I would like to emphasize that the software tools presented, Mega 11 and Eugene, as well as the online resources generalized as EMBL and NCBI, are only examples. There are many more tools out there, most of which are chosen by personal preference. What I am trying to say to you is that you do not have to use the tools the way I did. For example, I could have done the same phylogenetic tree online or with Eugene. Find the tools you're most comfortable with and try to get the most out of them. I hope that this lecture's practical approach has provided you with some useful tips. Thank you for participating and may your continued exploration be successful.